Dear friends, welcome to Biology Made Easy. We are today going to discuss gene mutation. So first we have to understand what is mutation. Mutation means a sudden change in the organism that is heritable. That means from generation to generation that should pass on. Mutation can take place in different ways. One is gross mutation, one is chromosomal mutation, the other is this gene mutation. Gross mutation means there will be change in the genome sets. Suppose one organism has 2n number of chromosomes, it will be 3n, 4n, 5n, 6n like that. That is known as gross mutation, or that is polyploidy, that we will discuss in a separate video. The second one, chromosomal mutation. In a chromosome, different genes are organized in a particular manner. If there will be any sorting between the genes, the first gene will go to the middle one will go to the last, reshuffling of genes. That is known as chromosomal mutation. That also we will discuss in a separate video. Today we will confine only to this gene mutation, which is also known as point mutation, and it is also known as molecular basis of a mutation. In this type of mutation, the change takes place due to change in a single nucleotide of the DNA. So this mutation can be classified in different ways. Here I have given one type of classification, samesense mutation, or you can call it a silent mutation. Second is nonsense mutation, and third is a missense mutation. What is the difference between these three things? Let us discuss it with examples. Here I am writing UG, 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 4 times I am writing this UG 4 times. Now we will write here U, C, A, G. So now there are 4 codons are there. Now if there will be any change in the first 2 bases of any of the codons, the change will be drastic. But if the change will take place in one of the 3rd bases, then it may give rise to these 3 types of mutations. Let us see one example. Suppose here U is there. Now suppose this U will be replaced by C. What will happen? If you will go through the genetic code, you will find these 2 are the synonyms for the same amino acid. That means the 3rd base is changed, but the amino acid doesn't change. This is known as samesense mutation or silent mutation. Mutation took place, but there is no effect on the organism. Now you see, now you come to the 3rd codon. Here U or C is replaced by A. So now the codon is UGA. And in genetic code you know that UGA is a nonsense codon. That means here whatever is taking place, the amino acids are joined together to give rise to polypeptides, that will be terminated. That means synthesis of protein will be incomplete. It will give rise to a dysfunctional protein. So that is nonsense mutation. Now you come to the 4th codon. In 4th codon, the 3rd base is G, UGG. So when U or C is changed to UGG, now the amino acid also will change. This represents tryptophan. So now whatever amino acid was there, that will be changed to tryptophan. So when the amino acid will change, the protein will change. This is known as missense mutation. Now for this I will give you a second example. All of you know that the hemoglobin has 4 polypeptides, 2 alpha and 2 beta chains. In one of the beta chains, at the 6th position, there is G, A, G, and the other one will be C, T, C. When at the 6th position, this sequence is present, this is the normal hemoglobin. Now suppose this will act as the template code for mRNA, then the mRNA will be GAG. This is in an mRNA. And this GAG will code for glutamic acid. It will code for glutamic acid. And in this case, the hemoglobin is normal, that is present in all of us. But suppose there will be mutation due to change in a single nucleotide. Suppose this adenine will be replaced by T. Now what will happen? When this will be replaced by T, then this will be automatically changed to A. So now the CTC originally was there. Now it will be CAC. When it is CAC, the mRNA will be, this A will be again replaced. Since it is A, here it will be U because it is mRNA. So now the codon is GUG. So see, previously GU, when present, it was given to glutamic acid and the hemoglobin was normal. Now due to mutation at a single point in a single nucleotide, now the GAG is converted to GUG and it will code for valine. That means at the 6th position of one of the beta chains, glutamic acid is replaced by valine. And what is the change? The normal hemoglobin is now converted to sickle cell hemoglobin. You see, by a single change in one of the nucleotides, what drastic change is taking place. This is again missense mutation because the amino acid changed. So this is also again another example of missense mutation. So I hope now you are clear what is samesense mutation, nonsense mutation and missense mutation. Now let us discuss the second type of classification. In the second type, the gene mutation or point mutation can be classified into frameshift mutation and substitution mutation. This frameshift mutation, again it may be deletion or insertion, and the substitution may be transition or transversion. Now in frameshift mutation, what happens? Suppose this is one DNA. This is the 5' and this is the 3'. And the sequences are, you can write anything out of the 4 letters, G, A, C, T, G, C, like that. Then, what will be the corresponding mRNA? The corresponding mRNA, G means here will be C, A means it will be U, C, G, T, A, G, C, and C, here it will be G. This is 3' and this is 5'. This is the mRNA. Now, if there will be deletion of one of these, means one of them will be removed. Any one, suppose this is removed, deleted. So what will happen? Automatically this will be deleted. Now see, previously the codons were CUG and ACG. But now what will happen? Now it will be CUG and then CG, then the next codon will come, whatever it is there it will come. Then the entire sequence of nucleotides will change starting from the point of deletion, unless there is a deletion of 3 nucleotides. If there is deletion of 3 nucleotides, then only one amino acid will be deleted, but all others will remain constant. But if there is 1 or 2, there is drastic change. Similarly, you come to the insertion. Suppose somewhere some nucleotide will be added. Suppose it will be added here another C. So here another C means here another G will be there. So now what will be the sequence? Previously in the mRNA it was CUG. Now it will be CUG. But you see the second one. Second one will be GAC. And the third one will be G, whatever are there. So you see again the sequence of nucleotide changes. That means starting from the point of deletion, the entire sequence of the nucleotide changes, causing a lot of harm. The new protein which will be synthesized, that may be non-functional, that may be lethal, anything can happen. Now, but what is the extent to which the deletion and insertion are affecting? You cannot imagine unless I give one example. So let us take one example. Ram has a red cap. In this sentence, each word has 3 letters just like the nucleotides. It is a meaningful sentence. Suppose there will be deletion. You can delete any one. Suppose this H will be deleted. Then what will read? Ram, then ASR, then EDC, then AP. Previously the sentence was Ram has red cap. Now what will it read? Ram, ASR, EDC, AP. That is only some jumbled up letters, no meaning. The same thing will happen in case of insertion. That means now you can imagine what is the drastic change that is taking place due to insertion or deletion of a single nucleotide. Right? Now come to the other one, the substitution mutation. In case of substitution mutation, there is neither deletion or insertion. Here simply one nucleotide will be replaced by another, and accordingly it may be transition or transversion. In case of transition, one purine is replaced by another purine, or one pyrimidine is replaced by another pyrimidine. Purine is replaced by purine, or pyrimidine is replaced by pyrimidine. So in that case we will call it a transition. But if a purine is replaced by any one of the pyrimidines, C or T, similarly G can be replaced by C or T, or vice versa. The C can be replaced by A and G, or the T can be replaced by A G. When such type of changes take place, this is known as a transversion. Again I am repeating, when a purine is replaced by a purine or a pyrimidine by a pyrimidine, it is known as transition mutation. But when a purine is replaced by a pyrimidine and vice versa, it is known as transversion mutation. Now, what is the example of this transversion mutation? Now here also you can give the example of sickle cell anemia. In sickle cell anemia, in the original hemoglobin, it was GAG and the CTC. Now, in mutation, this A was converted to T. So, A was adenine, that was a purine, and thymine is a pyrimidine. So, a purine is replaced by a pyrimidine. So, what type of mutation it will be? Transverse mutation. That means this sickle cell anemia is an example of transversal mutation. It is also an example of missense mutation. So, I hope you must have understood the whole thing of this gene mutation. But for clear understanding, first you go through the genetic code, then you read this gene mutation, so your understanding will be 100%. Now, I will give you few questions. Number one question. When a single nucleotide changes, it leads to A choice gross mutation, B choice chromosomal mutation, C choice gene mutation, D choice none. Then come to another question. When in the codon UUU, when in the codon UUU, the third base is replaced by C, the third position will replace by C, then it will be A, first missense mutation, B, samesense mutation, C, nonsense mutation, D, none of the above. After going through the video, you will be able to answer all these questions. I hope you have understood it perfectly. So, thank you very much and wish a good day.